Hello everyone, I'm Nicoleta Spinu, currently a researcher in Mark Cronin's lab in Liverpool, England. I was responsible for the project 12, which focused on the development of quantitative adverse outcome pathways types of computational models. I believe everyone wanted this meeting to have happened in person rather than online. I must admit, it feels quite strange to register our results and to present to you virtually. I'll do my best. If you have any questions or you want to discuss anything in particular, please get in touch with us. I'll start this presentation by explaining what a QOP model represents, especially as there is no regulatory guideline to help with the development and evaluation of such models. Luckily, there is an OECD guideline on the integrated approaches to testing and assessment, abbreviated as IATA, that describes a QOP model as a quantitative understanding of what magnitude of change is required in the upstream key event to evoke a magnitude of change in the downstream key event. To put this into other words, a QOP model represents a sequence of causal effects that will take place at different biological levels, starting from molecular level where a chemical or any other type of stressors such as environmental factors, genetic predisposition, that will lead to effects at cellular, tissue, organism, and population level. Besides this qualitative mechanistic description that a QOP model requires, it is also about a computational approach that to be utilized, and especially the use of such computational approaches are for defining these tipping points, also known as magnitude of change. And these tipping points describe the transition between the upstream key event to the downstream key event in terms of time and concentration. Therefore, a QOP model requires kinetic and dynamic type of information as well. At present, there are 3 computational approaches proposed to quantify NLP. It includes a semi-quantitative quantitative weight of evidence approach that quantifies the level of maturity, the level of confidence in the qualitative LP based on the Bradford-Hill considerations and also using a scoring system, for example. Of course, this QOP model can also incorporate other type of criteria, not only Bradford-Hill criteria. The other 2 approaches are the probabilistic and mechanistic ones. Interestingly, in a recent review that we've been conducted, one of the conclusions was that the scientific community expects that the QOP model will be about probabilistic and mechanistic approaches, where we can define those tipping points in terms of probabilities, for example. For the past 3 years, we've developed 3 methodologies. With some of them you might be familiar with, for example, the development and analysis of an LP network. I'll present each of them, and I have to mention that as a case study, neurotoxicity was chosen. And I'll start with the first methodology, which was about the development and analysis of a network of linear LPs. It is composed of 4 steps, and it starts by defining the purpose. In our case, we wanted to assess neurotoxicity. It is followed by defining the set of criteria for which we want to evaluate each independent linear LP. The third step is about collecting those linear LPs from the LP wiki knowledge base of the OECD. This database has been recently updated with a much more user-friendly interface. And besides that, this step already can be automized and not necessarily to extract manually those linear LPs. And the final step consisted of generating the network and also analyzing the topology analysis. While this step can be conducted, for example, in Cytoscape software, which contains several types of apps inside, and one of them can be used specifically for analyzing an LP network in order to identify common key events across the network. Also, Cytoscape contained another important app, especially for our community, which is called LP Explorer, and this work is currently available there. This is the resultant AOP network for neurotoxicity following the methodology that we've developed and proposed. It managed to combine 9 linear AOPs out of 12 linear AOPs that were available at that time. And it describes 3 adverse outcomes: Parkinsonian motor deficit, neurodegeneration, and impairment of learning and cognitive function, the decrease of this cognitive function. Besides that, the molecular initiating events are included at present in a ESR model developed in collaboration with the lab from Mario Negri Institute Milano. The squares in blue, colored in blue and filled with blue, were the key events identified as common key events across the network that linked multiple linear AOPs. For example, the reduction of brain-derived neurotrophic factor, the decrease in synaptogenesis, or the decrease in neural network formation. Now, based on this work, we developed 2 more methodologies. One of them focused in quantification of a simplified version of an AOP network with the focus on developmental neurotoxicity, and the second one represented an attempt to quantify a linear AOP, and more precisely the AOP-ID3, with the focus on the Parkinsonian motor deficit as an adverse outcome. I'll continue with the second methodology, which was about quantification of a simplified version of the AOP network for neurotoxicity. Several common key events were selected in order to represent better the AOP network. The selected common key events were based on the topology analysis, as I've shown in the previous slide, but also based on the domain knowledge, so based on an expert panel. This simplified version of an AOP network served as a basis for data collection, as well as to define mathematically regression functions. We wanted this quantification to be done following a probabilistic approach, and in that sense, to follow the Bayes theorem. The prior was defined as weakly uninformative because we wanted our model not to overfit the results. The prior means what we consider the distribution of the data to be before seeing the data, so before the model actually analyzes the data. What is of interest out of such probabilistic model is this posterior distribution. So each compound independently from our dataset was evaluated for this biological path, and a posterior distribution was computed. What is also interesting to mention is that these probabilistic approaches are recently increasingly applied, and that's because of the power of computational capabilities. If to do mathematically this normalization constant, it gets very complicated. But with Markov chain Monte Carlo algorithm, for example, this can be efficiently done. Also importantly is that with this conceptual framework where we wanted to quantify a simplified version of an AOP network, another reason why we chose the Bayesian approach is that we wanted to be able to incorporate the diverse type of evidence that is available, especially when we know that developmental neurotoxicity is not well studied, not yet well studied. And here I present to you the type of data that was collected, and as an example, I took the bisphenol A. Now, the details included in silico type of information, information about lipophilicity, if a chemical crosses or not the blood-brain barrier, and details about P-glycoprotein, if a molecule activates PGP, inhibits, or acts as a substrate. And this was possible with the help of Liadis Moralagares, who developed an in silico model to screen molecules for PGP. Other important details were to collect important information about each of the key events. For example, reduction of PDNF. It was based on the peer-reviewed literature, and this allowed us to classify a compound into a binary system, yes, no. For the decrease of synaptogenesis and decrease of neural network formation, 2 in vitro studies are available, and information about those toxicity assessments that has been conducted were included, and a classification of yes, no. And finally, for the adverse outcome, the classification was also binary one, and it was also done based on a systematic review, the peer-reviewed publications. Here we see the technical specification of the Bayesian model. I'll not go into details. However, I have to mention a few important aspects. The Bayesian model followed a hierarchical type of modeling in order also to avoid the overfitting of the model. And for that purpose, we defined a common mean and standard deviation that describes the entire data set. We combine the data with the parameters in order to describe each of the key events independently, and at the same time to follow the structure of the LP network. And hence each chemical was screened across this type of model and has a probability associated for each of the key events independently and the adverse outcome of DMT. Importantly, it is to mention that the dataset contained missing values. That's also because the dataset was composed of both organic and inorganic compounds, and also we had missing values across both predictors and outcomes of the models. For example, those 2 in vitro studies didn't test the same list of compounds, and that's one of the reasons why we had missing values. And the missingness we've computed as well in a Bayesian way. So the missing values that were present in predictors were computed from the prior distribution, and the missing data for the outcomes were computed from the posterior distribution. Here we see the results of the distributions of the predicted